Operon model of prokaryotes. What is operon? Operon is cluster of bacterial genes along with a promoter region that controls the transcription of those genes. In prokaryotes, operon includes structural genes, the promoter, and the operator. In this diagram, we can see that these are the structural genes, gene 1, gene 2, gene 3. There is a operator region and there is a promoter region. So collectively, structural genes, operator, and promoter is called as bacterial operon. Another diagram of the organization of a bacterial operon. Here we can see that these are the structural genes. Here is the operator region and here is the promoter region. So collectively, it is called as operon. Here we can see that this all is operon. Components of operon. There are three components of the operon. The first one is repressor. The repressor gene encodes a repressor protein that can also be called as regulatory protein. The repressor binds to operator. Binding is regulated by the presence or absence of effector molecules that can be inducer or that can be repressor. Other components of the operon, they are promoter and operator. The promoter is a site of transcription initiation for the structural genes, means from where there is the production of the translation of the structural gene starts. That region is called as promoter. Transcription of the structural genes is regulated by binding of repressor to the operator. In this diagram, we can see that this is the diagram of lag control system or bacterial operon. These are the genes which are called as structural genes, or they are called as coding region. Here we can see that here is the operator region, then genes for the repressor protein, then promoter for I-lag gene, that actually promoter of this I gene that produces a regulatory protein, and this regulatory protein controls the expression of the genes, whether by inducing or by repressing. There are two types of the operons. One is called as inducible operons, in which transcription is initially the transcription is off and needs to be turned on. So to induce the transcription, that is the reason it is called as inducible system. While in case of repressible systems, transcription is normally on and needs to be turned off. That is the reason it is called as repressible systems. There can be positive and negative control mechanisms. Regulatory genes, they encode the proteins which are called as regulatory proteins. They regulate the expression of other genes. So those genes, they actually regulate the mechanism either in the form of positive control or in the form of negative control. In case of positive control system, the product of the regulatory gene is required to turn on the expression of the structural genes. In case of positive control system, the regulatory gene products are called as activators because they activate the transcription of the structural genes. While in case of negative control mechanism, the product of the regulatory gene is necessary to switch off the expression of the structural genes. The regulatory gene products are called as repressors because they repress transcription of the structural genes. So control mechanism can be in the form of positive control mechanism and it can be in the form of negative control mechanism. So here is the example that the lag operon is repressed because here we can see the lag repressor binds to the operator. When it binds to the operator, there is no translation of the structural genes. Here is lag operon induced because due to the presence of the lactose, now the repressor will not bind to the operator region. So there is the translation of the structural genes. We will call it as lac operon is induced. If there is glucose low inducer is present, then the promoter is activated. When the promoter is activated, the RNA polymerase is not blocked and there is a translation, so the messenger RNA will be produced from the structural RNAs. When there is glucose high inducer absent, promoter not activated. When promoter is not activated, then the repressor binds to the operator region, and so there is RNA polymerase is blocked by the lac repressor, so there is no translation of the structural genes. What is tryptophan operon? The tryptophan operon is negatively regulated by the tryptophan repressor protein. The tryptophan repressor binds to the operator to block the transcription. Binding of the repressor to operator requires a co-repressor, which is actually tryptophan. Low level of tryptophan prevents the repressor from binding to the operator. If the tryptophan is absent, then the promoter is activated. Here we can see that there is no tryptophan, so promoter is activated. When the promoter is activated, so there is the translation of the structural genes. While on the other hand, if tryptophan is present, then the promoter is repressed. When the promoter is repressed, so it will block the function of the RNA polymerase and there will be no translation, there will be no transcription of the structural genes. So if we conclude that what is operon model, prokaryotes, they have operon models which actually consist of structural genes, operator region and the promoter region.